basically the flow of genetic information, with emphasis on DNA replication. And hopefully it's, like I said, a lot of review from Bio 161. Now, this is an emphasis on bacterial genetics, and so the proteins and many of the things that are involved hopefully will be new. The concepts on this particular slide start with the central dogma, the pathway from DNA to RNA to protein. And it's pretty much conserved in all cellular life forms, but we know that retroviruses do not follow the central dogma, which is interesting because I always think about that. If the initial studies on the central dogma weren't done on DNA-based E. coli phage, and instead were done on retroviruses, we would probably have been set back a little bit in terms of our understanding of the central dogma. So just by way of comparison here. Anyway, here's a typical retrovirus infection cycle, just to show you this reverse transcription process. And again, I don't need you to know all the details of this. If you want to know about this stuff, you could take Micro 320. But here's a retrovirus like HIV, and it's an envelope virus, and so the virus will undergo a membrane fusion. In that particular case, that membrane fusion event will allow the capsid of the virus to enter the host cytoplasm, and then through a series of reactions, including acidification and so forth, the capsid breaks down, as you see here, and that releases the viral RNA. The polymerases that retroviruses have are RNA-dependent DNA polymerases. So they're looking for this RNA template. And so the RNA template is bound, and the polymerase actually makes a DNA copy of that RNA, a single strand, and then it turns around and makes the double strand. So you end up with a double strand of proviral DNA, and then this DNA can integrate into the chromosome of the host. Again, I don't need you to know all those details, but I do want you to understand the exception to the central dogma. Obviously, DNA stores the genetic information, and it's passed from generation to generation. So one of the things that I wanted you to do is, obviously these next few slides are DNA replication, but the easier way to look at this is through a video. And so I found a pretty good video that summarizes all this stuff without me having to go through these boring slides. And so it's this particular video, and it's
involves a number of proteins coming together in a complex machine. DNA replication begins at a single defined DNA sequence of...
So again, I'm not going to play the whole thing, but you have the URL here on this slide. And so again, that'll hopefully help you with these next few slides. This one's just showing you the concept that's indicated in this slide about semi-conservative DNA replication. And so in that particular case, you have the parental helix is being unwound, and so that creates two replication forks. Each replication fork then consists of a parental strain and then the purple daughter strain, and that shows you the concept of semi-conservative replication. The concept here is that there's the two main sites, the origin of chromosomal replication, or OREC, and at the other 180 degrees away is the termination sequences, And so then you would get bidirectional replication heading in those two directions, as you see here. So here's one fork and here's another fork. And at the end, the replication machinery bangs into the ter sequences, the termination sequences that essentially stop the helicase. So this particular slide from this other lecture tends to be a little bit informative, and so I threw it out there just so you could see it a little bit better. But here's the OREC. Here are the replication enzymes, the replisomes, if you will, heading bidirectionally, and then they bang into these termination sequences, which are bound by TUS monomers. So these TUS proteins will bind to these termination sequences, and when the replication fork gets here, it starts banging into them like a speed bumps, if you will, and eventually it causes the replication forks to arrest or stop and then fall off. And so we get termination of replication, rather than having these forks just kind of keep moving around the circles indefinitely. We don't want that to happen. So termination is really important and highly regulated. This slide just gives you an example or an example of each protein and then the function of the components of the E. coli replication machinery. And so the main ones are DNA A, DNA B, gyrase, the single-stranded binding proteins, DNA C, DNA primase, the polymerase 3, which is the main replication enzyme, polymerase 1, which fills in the gaps, and then RNase H, ribonuclease H, and DNA ligase. And of course, then the replication termination proteins, the test proteins, for example, and then the role of topoisomerase. So I want you to focus on those things that I just mentioned, and then look at the video and kind of cross-reference how these things work. This particular slide just shows you the main enzyme, DNA polymerase 3, the main replication enzyme, at least in bacteria. Remember that eukaryotes have maybe a dozen or so polymerases, whereas E. coli has approximately 5. And I just kind of indicated them here. 2, 4, and 5 polymerases are repair polymerases. They are able to synthesize DNA over lesions. If there's any kind of damage to the DNA, they can synthesize over it, but they make a lot of mistakes. So the trade-off there is, well, you're going to get some replication across the banged up region of DNA, but odds are it's going to have some errors in it. And then polymerase 1 obviously is also repair and replication, filling gaps and so forth. And then of course polymerase 3 is the major replication enzyme. So this thing is made up of a few different components. The first one is the first thing is the beta clamp that you see here that forms the ring around the DNA. This little donut essentially keeps the polymerase, the core enzyme of the polymerase, in contact with the DNA, and theoretically the DNA would be threaded through here. The core enzyme consists of 3 proteins, alpha, epsilon, and theta. Alpha has the main polymerase activity. Epsilon has the exonuclease activity, and theta seems to be for assembly, presumably binding of this to the TOF protein or tau protein. And so that's more of a structural thing. But the exonuclease activity that you see here is really important. Obviously, that gives polymerase 3 the ability to, if it makes a mistake, to back up, chew off the mistake, and then lay down the correct base. Here's DNA helicase. That's responsible for DNA B or DNA B helicase that's responsible for strand separation. And so again, you see this information here, that the epsilon fragment is responsible for 3', 5' proofreading. So again, the polymerase is going to lay these bases down 5' to 3', and then if it encounters a mistake that it made, it'll go backwards 3' to 5', fix the mistake, chew it off, and then put the right base down, and then turn around and continue in the 5' to 3' direction. So I always like to throw these things in here just as a way to give you context or some interesting facts about why would we need to know about this kind of stuff. And and one reason why we would need to know about it is how to interfere with the process if we want to block microbial growth. And so quinolone antibiotics are a good example of that, specifically ciprofloxacin, and it's abbreviated Cipro. The antibiotic will bind to topoisomerases. So here you see two replication forks, and here's a DNA polymerase complex and another one. And so these things are moving along, replicating. And the topoisomerases are required to create strand breaks, and so in the process of unwinding the DNA, you have to make breaks in the DNA and then reform those breaks in the process of unwinding. And so the quinolones bind to these topoisomerase, DNA gyrase, and prevent them from making this from completing the strand break reseal process. Basically, they catch the topoisomerases in the strand break phase, and so that's why you see the drawing here with 2 strands of broken DNA, and then with the quinolone antibiotic stuck to them. So they can't reform, they can't reseal the DNA. And as a result, replication arrests. Don't worry about this stuff here about ROS formation and protein independent and protein dependent. That's not important. I just wanted you to see this more than anything, how quinolone antibiotics interfere with topoisomerases to prevent replication, DNA replication specifically. And this slide is basically intended to go along with the video that I just initiated previously. And so you can use this and the video to understand the process and how each of the components like helicase and primase and SSBs work to facilitate DNA replication. This slide gives you the extra concept of Okazaki fragments. And so the video mentions Okazaki fragments and how they're made and then how they are basically sealed in the lagging strand. And so again, here's more information about the fork. So this slide, but the others can go along with the video to help you study the material. And then here's the last phase, the termination concept. So I've already talked about the role of termination sequences and the TUS proteins, but this is showing you a way that topoisomerase can temporarily break the DNA so that the strands can ultimately separate. And so that's pictured here. So at the end of replication, you basically get two links in a chain that are formed. But you can't have that, right? Because this chromosome has to separate from this one and then move to a new bacterium. Each of these has to move to a new bacterium, but they can't like this. And so there has to be a double-stranded break, as you see here, and then these things have to segregate into different daughter cells. So it's not pictured, but if you look at this particular slide, that would be happening here, where this chromosome would be linked to this one, like links in a chain. And then ultimately, you'd need topoisomerase to break those links and facilitate segregation of these into new daughter cells.